Welcome back, welcome back, welcome back to science. So, today we're going to talk about spermatogenesis. Ultimately, it's the formation of sperm. You're going to see some words that are going to have like spermatogonia. And honestly, this is going to really look at the process of meiosis and the way it's working in human gamete cells, specifically in the testes to make a sperm cell. So, there's a type of cell called a sustinator cell. Uh, this is a type of Sertoli cell. So Sertoli is the more common name, and so instead of the sustentacular, these are going to be large cells. They're going to be elongated, and eventually, whenever you have the actual formed sperm cell, it's going to work its way into the lumen that's going to be in the seminiferous tubules. These are going to be spermatogenic cells, meaning that they're going to form sperm. There's a series of named cells in the process of sperm cell formation. You're going to start with spermatogonia. These are going to give rise to the primary spermatocytes. That'll give rise to the secondary spermatocytes, giving rise to the spermatotids, giving rise to the spermatozoa. Meiosis is the process by which we go from diploid to haploid, going from 46 chromosomes in an original cell to 4 haploid cells, each having 23 chromosomes in them. So, the development of spermatids into sperm is called spermatogenesis. Genesis is the creation of, so this is the creation of sperm. So, spermatogenesis combine the processes of meiosis and spermatogenesis. 4 sperm cells are produced, and from the formation of through meiosis. So, let's take a look. We have the primary spermatocyte. Notice that we have 46 chromosomes. Here we're forming the tetrad. This area is going to be separating. These are replicated homologous chromosomes. So this is a homologous chromosome to this. This is a chromatid, 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 chromatid. This process of the first meiotic division is separating the chromatids. You can see that this cell and this cell, even though it looks like they're the same, the original material, let's say that's from the mother and this is from the father, that they're separating through the process. If you don't understand what I'm saying here, feel free to go back and look at the video on meiosis. So we have the secondary spermatocyte, which is the halfway point. Eventually, we go through the second mitotic division, and eventually, we're going to have 4 different spermatotids. A few things to look at with a spermatotid. We have the head, but notice that there's an area on top. This is an acrosome top. You have a body, and in that body, you're going to have a mitochondria, and from that, you're going to have the actual flagella. That is kind of the characteristic marking of a sperm cell. So during spermatogenesis, the spermatogonia replenish themselves and give rise to spermatocytes. Those develop into sperm. So as the stages of spermatogenesis continue, the developing sperm migrate from the outer edge. And so imagine a tube with a hole like a garden hose that is cut. Now you're looking down the circular part of that. The mid region of that is going to be called the lumen. That's just an opening. The outer side is going to be the most immature cells that are giving rise to more mature cells as it moves inward. And eventually, once the cell has come to the point of full maturation, it is in the center part of that lumen. So the sustentacular cells support the entire process, housing and nourishing the sperm cell. So this is showing the outer wall. This is the opening to the middle of that. Once again, you're looking at the sides of one of these tubes, and as you look down the region of that around the edge, you're going to have more and more mature of the cells as you go towards the lumen. Sperm structure, as I pointed out a minute ago, you have a head, which is going to contain the chromosomes, but it's also going to have an acrosome cap. We'll look at the acrosome whenever we look at the egg cell that's fertilized. That process of fertilization, that's going to, it requires the acrosome tip, which has enzymes in it, that's needed to actually get through the outer layers of an egg cell. Without that, a sperm cell wouldn't be able to give its DNA to that egg cell to get true fertilization. All right, down from the head, you have the midpiece, where you find the mitochondria. Below that, you have the flagella. Flagella is going to have whip-like undulations. That's going to cause it to be propelled to the point of finding, theoretically, hopefully, by theory, finding a sperm, the egg cell. All right, so here are some sperm cells that you see through the microscope. All right, guys, that's going to finish up that subsection. Be back soon.